Hi, students. This is social behavior in animals, and we're going to be talking about neuroendocrine systems and behavior today. So I'd like to share my screen with you, and we'll work our way through the lecture we are going to talk about. So I'm sharing my screen with you now, and this is our lecture for today, Neuroendocrine Systems and Behavior. We're starting to think about the causes of behavior and the ontogeny of behavior in the next set of lectures. So we're really focusing on proximate questions about behavior. And it's difficult to kind of talk about the nervous system, the endocrine system, genetics, and focus on all types of behavior. So we're going to narrow this down. Today I'm going to focus on oxytocin, vasopressin, and bonding behavior, a social behavior we see in prairie voles. And we'll start then with some real basics, but move into some details of some of the research done in this field. And so we're thinking about the factors that influence or contribute to behavior. We're, as a field, getting away from this notion of nature versus nurture and recognizing that there's a really complex set of relationships between nature, nurture, and behavior. We'll talk about this again in our next lecture, but this diagram that is in front of us, it shows us a little bit about the relationship between genetics, physiology, and behavior, and the environmental influences that can affect behavior as well. So we've got behavior on the right, and we recognize that, oh yeah, there are different parts of behavior from an embryo to an adult stage, and behaviors in childhood can influence, or infancy can influence what's happening in adulthood. And when we look back at the genetic and the physiological components to behavior, we can start way back with the genes. Genes are basically codes for the building of proteins, and proteins regulate all sorts of things inside our cells. It says our cell metabolism and response here, including our nervous system, and how our nervous system operates influences directly what our behavior is, right? Our nerves control what behaviors we make. And in addition to all that, there are influences from both our internal and external environment. So things like our diet and our hormones control and regulate which genes are expressed, which proteins are made, which behaviors are performed. Our diet can have a big influence also on which genes are turned on and turned off in our genome. And our external environment is also important. Things that we're exposed to influence us. Our early childhood, we know, influences our behaviors. I also believe that there needs to be a link between behaviors and genes. Our behaviors actually regulate our genes, and we'll talk more about that later. We're going to focus on a little bit on the endocrine and the nervous systems today, and how they interact to facilitate social behavior. There are two links here that, if you're interested, you can learn more about the endocrine and nervous system. We're going to do a quick review of both of those just to remind ourselves about what they're like. The endocrine system is our hormone system, right? Our endocrine system is made up of organic chemical messengers that are released from targets. Just a second. I'll be right back. What did you see this? Alrighty. So the endocrine system is a set of organic chemical messengers that are produced by specialized glands in our body, and they're released by these glands and into the bloodstream. And from the bloodstream, they travel around and they approach and reach targets. Targets are chemicals that, are cells that actually have receptors for these hormones. So here's a picture of our bloodstream with little blue dots. Those are the hormones, and they target certain cells. They attach to certain cells and because of receptors on those cells. And when they're attached, they cause changes in the behavior of those cells. They may trigger changes in which genes are turned on or turned off in the cell. They may trigger in the nervous system, for example, which neurotransmitters are released or the rate at which they're released. And so you can see over here a list of a number of basically all of our different glands that make different kinds of hormones and release them into our blood. Now, the endocrine system tends to mediate long-term processes like growth, reproduction, metabolism, and they tend to influence the likelihood of behaviors happening. So, for example, if we're thinking about androgens, androgens are made in the testes. They're also made in the adrenal glands. Androgens include things like testosterone, and androgen targets are all over the place. Androgen targets can include things like the testes, cells in the testes that help to make more testosterone, and cells in the testes that make sperm. They target muscle cells. We know that, at least during human adolescence, an increase in androgens like testosterone lead to an increase in skeletal muscle, and also a whole suite of secondary sexual characteristics that develop during that adolescent period. Things like skin cells can be affected, and in fact, the androgens can target regions of the brain as well, right? Certain parts of the brain have receptors that respond to testosterone and other androgens and help to regulate aggression and sexual behavior, for example. We say that the endocrine system influences the likelihood of behaviors happening. So, for example, if we're thinking about testosterone, testosterone's really important, we think, in territorial behavior in songbirds. Male songbirds have increases in testosterone levels during the breeding season, and we also see an increase in their vocalizations in the songs that they make, and an increase in aggression. And there's a correlation between increase in androgens and increase in aggression toward rivals. Androgen increase the likelihood that these birds are going to be more aggressive. They're going to sing more songs during the breeding season. Okay. The endocrine system tends to operate in a kind of slower way than the nervous system. It might take hours or days or months for the endocrine system for the effects of the endocrine system to show up. The nervous system operates in a different way. The nervous system sends messages through interconnected neurons, which are shown up, a set of neurons are shown up here. And when we think about the nervous system, we think about these messages as being digital, all or none messages that are electrical impulses that are passed from neuron to neuron to neuron. And they're passed usually through a long extension we call the axon. And electrical signals are essentially changes in the membrane potential of the cell. If you're not a neuroscientist, that's okay. You don't need to understand this completely, but the basic idea is these cells send nervous signals or electrical impulses that are very rapid down the length of the cell, and they arrive at the next neuron or at a muscle cell or a gland or something like that. Here's a picture of the end of one neuron and the start of another. This is the dend right here. There's a gap. And as the electrical potential gets sent down the length of one neuron, at the end of that neuron, it may release neurotransmitters, chemical signals that the next neuron responds to, that the next neuron can receive and change its behavior when responding to it. Neurotransmitters like dopamine and norepinephrine and epinephrine are some examples that you'd probably be familiar with. So the nervous system tends to operate in a rapid, all or none way that has very discrete onset and offset. The nervous system has different parts, right? The brain and the spinal cord represent the central nervous system. The peripheral nervous system is everything else. There are autonomic nervous system. That's the autonomic nervous system would control things like involuntary functions like our heart rate and our digestion. The somatic nervous system will control things that are more voluntary, like our movements, our muscle actions, those kinds of things, right? Okay, so we're interested in this, like the relationship between hormones and the nervous system and how hormones, nerves contribute to social behaviors. D here is a comparison of the endocrine to the nervous system. And you can see some of the basic similarities and differences between our nervous system in a whole suite of things. So, for example, the nervous system is very rapid, the endocrine system is quite slow. Nervous system uses electrical impulses, the endocrine system uses chemical messages sent through the blood. Yeah. All right. Now we're ready to really think about oxytocin and vasopressin and their role modulating social cognition and behavior. I chose to think about this particular topic because I find it interesting and exciting, and it connects a little bit of endocrine system with a little bit of the nervous system with a little bit of behavior. And so we're going to think about oxytocin and vasopressin. And both of these are hormones. They're actually little tiny proteins called neuropeptides that are short and small, and they're made in the hypothalamus, which is a part of the brain that is actually involved in making a whole suite of different hormones that help to regulate lots of different things like appetite and sleep and sex, and reproductive behaviors in general. And here are some pictures of what oxytocin and vasopressin look like. They're actually made up of, in humans, 9 amino acids. And vasopressin and oxytocin are very similar. Now, they're made in the hypothalamus. They're released actually in a gland called the posterior pituitary that sits at the base of the brain. This is a human brain that we're looking at here. And then they're released into the bloodstream, where they have all sorts of targets. Here's a picture on the right of some of the brain targets. So oxytocin and vasopressin target some structures in the brain, in the central nervous system, in addition to targeting lots of other places in the body. It turns out researchers believe that oxytocin and vasopressin and their homologs, or molecules that seem to be evolutionarily related, have been around for a long, long time. And on the left, we have a chart that shows some of the different neuropeptide hormones that have been found in really different kind of animals. And they seem to play a role in modulating reproductive behaviors in a lot of different animals. In mammals, there seemed to be mostly a sexual dichotomy of function. By that, we just mean that males and females, in males and females, these hormones act differently, right? In females, Oxytocin has long been implicated in labor. It causes contractions of the uterus, which facilitates the birthing process, and it's involved in lactation, particularly in the letdown of milk, so that mom can secrete milk for baby to suckle. It also seems to be really important in bonding behavior between mom and baby, between the maternal infant bonding behavior. We'll remember that. Vasopressin, on the other hand, seems to be involved in male mammals seems to be related to, at least, territorial behavior and sexual behaviors like erections and ejaculations. And entirely separately, it seems to be involved in water reabsorption at the kidneys. Right? It's got all sorts of functions. We're interested in thinking about a particular mammal called the prairie vole and a particular social behavior called pair bonding, and thinking about the role of oxytocin and vasopressin in both. So here we go. Let's give you a little background on prairie voles. So first of all, prairie voles live in North America. Here's their home range over here. Oh, they might live in Michigan, maybe. In any case, they live in prairie habitats in burrows, and they are territorial, so they tend to be aggressive toward each other. In the wild, they live very short lives, somewhere around a year, maybe a little longer. In captivity, they can live 2 to 3 years long. They tend to feed on vegetable things, so greens, roots, seeds, bark, stored or cached foods in the winter. And there are zillions of predators for these little buggers. Owls, hawks, ravens, snakes, coyotes, foxes, weasels, badgers, you name it. Everything attacks these little guys. Their reproductive behavior is pretty interesting. Their mating system is particularly interesting. They are what we would call socially monogamous. Socially monogamous. They form pair bonds during the breeding season, and these are lifelong pair bonds as far as we know. And in these pair bonds, the males and the females are going to participate in parental care. So they mate, and then gestation is short, about 21 days. The prairie voles tend to have about 4 pups per litter. And during this gestation period, the males and females will build a nest together. And then once the pups are born, they will take care of the pups together. They will raise them together. And after the pups leave the nest, they will continue and maybe breed again and have another litter. And so this pair bond that forms is a lifelong pair bond for most prairie voles. They may have multiple litters along the way. Okay. Close relatives of the prairie voles are meadow voles and montane voles. Meadow voles live more easterly. Montane voles live more westerly, their home ranges at least. But both of them are promiscuous, we might say, or polygynous species. In other words, meadow voles and montane voles don't form these pair bonds. The moms take care of building the nest and all of the parental care and defense of the young. Males typically breed and leave. All right. So the interesting thing is, well, what's the difference between prairie voles and meadow voles or montane voles? Prairie voles are really weird, forming pair bonds. It's unusual. It's an unusual mating system. And so that's what we're interested in. What have we learned about prairie vole pair bond formation? And so let's take a look. I've got a few things that you'll see kind of contribute to our story, our knowledge about oxytocin and vasopressin, and about pair bonding. But it's not a finished story. we still have a lot to learn about the relationship between these two hormones and social pair bonding formation. So this is what some early studies on oxytocin. So we'll start with oxytocin. Oxytocin is this hormone that we typically think of as contributing to female behaviors, female reproductive behaviors, right? And so back in the 1980s or so, researchers started making oxytocin injections into females of different species and injecting the oxytocin into different brain regions and trying to identify how did that contribute to changes in the animal's behavior. And so they injected rats, female rats, actually virgin female rats, with oxytocin, and oxytocin injected rats suddenly became very maternal. They started engaging maternal behaviors that most virgin females would not do. Researchers also injected sheep with oxytocin and found very similar things. This injection of oxytocin induces nurturing behaviors, so that's pretty interesting. And the evidence is pretty strong that oxytocin seems to be related to a whole suite of maternal behaviors. But with prairie voles, researchers started looking at pair bonding, the formation of pair bonds and the maintenance of pair bonds, and asking the question, how does oxytocin play a role in that? Does it? That's the question, right? And so researchers added or injected oxytocin into different parts of the brain. And in particular, they injected oxytocin into part of the basal ganglia that is involved in the reward system. And this is called the nucleus accumbens, or accumbens. And this nucleus has been now kind of studied quite well, and researchers are pretty sure that oxytocin here is involved in the formation of partners or bonds. So researchers have done a whole suite of things. In this particular study here, researchers can take a female prairie vole, insert oxytocin injections into that prairie vole, and that stimulates bond formation much more quickly than without that injection of oxytocin. And in a second study, researchers said, all right, well, let's look at let's look at what these animals do after a bond has formed. So now we've got this, we think a bond formed, the males and females mated, and 6 hours later or 24 hours later or 48 hours later, will the female actually show a preference for this mate, for this male that she mated with 6 hours ago or 24 hours ago. And so what they'll do is a preference test where they put the female in the middle and they put two tethered males on sides of the cage, a stranger and a little male who is the partner, and then ask, hey, how much time does she spend with the stranger versus the partner? Does she spend more time with the stranger or the partner? And oxytocin injections lead to a few things. First, they lead to the faster formation of partner preferences and a stronger formation of partner preferences. Then researchers said, hey, let's use some sort of oxytocin receptor blocking chemical. Let's block the oxytocin receptors in the brain of females and then ask the question, do they form partners or do they show a partner preference? And they did that. They blocked oxytocin receptors and showed that, nope, that inhibits partner preference formation. So oxytocin receptor blocking prevents partner preferences. All right, so oxytocin seems to be oxytocin, at least injected into certain regions of the brain, the nucleus accumbens for sure, seems to be really important in forming bonds and in maintaining those bonds. And that's interesting because that hadn't been shown before. Maybe researchers thought that prairie voles have sort of co-opted oxytocin, which played a role in maternal infant bonding. Maybe they co-opted it for pair bonding. Maybe it's now got another function. And so they started looking also at the receptors in the brains of voles. What are the oxytocin receptors in the brain? Where are they? And what do they look like? So it turns out that there are regions of the brain that whose neurons have oxytocin receptors, whose neurons are going to be sensitive to oxytocin. And the particular region that shows a really high density of receptors for oxytocin in the brain of prairie voles is the nucleus accumbens. The nucleus accumbens is in the basal ganglia. It's in the reward circuitry. It's a part of this bigger network of neurons that are involved in reward, in learning about reward, in responding to reward, and actually also learning about aversive stimuli. But you can see in maternal prairie voles, that's this diagram over here, the nucleus accumbens has a very high number of oxytocin receptors compared to a non-maternal animal, right? And so researchers decided, hey, let's mess around with oxytocin receptors, and let's make some changes and look at pair bond formation when we do some little surgeries and mess with oxytocin receptors in the brains of voles. And so here's what they decided to do. Yes. Okay. Hang tight. Let me turn my page here. All right. So here's what they did. They did several surgeries. One was a sham surgery. That's what a sham comes from here. And they did two other types of surgeries in which they basically introduced the oxytocin receptor gene into the nucleus accumbens of prairie voles or meadow voles. That's what the OTR stands for, oxytocin receptor gene. And they introduced a totally control gene, a gene that doesn't contribute to oxytocin receptors at all, into the brains of other prairie vole females, right? And then they looked at, partner preference, basically. They did these partner preference tests. They looked at what percent of the time are you in how much time are you in close proximity to your partner after a really short duration, like 6 hours of being with a partner, and the percent of time with with your partner. All right, how much of the time are you spending with your partner, right? And it turns out that as you can see, if given only a short amount of time with your partner, control animals didn't show a preference. They really didn't. But these animals that were given oxytocin receptor genes inserted into the nucleus accumbens, which basically means there's a lot more receptors in this set of neurons, a lot more receptors for oxytocin. We see an explosion of behavior, right? We see this partner pair bonding behavior in much higher rates. So the presence of these receptors in this nucleus in the reward system of the basal ganglia of the brain means partner preference. Meadow voles are kind of interesting, and this sort of was unexpected. Researchers totally predicted how the prairie voles were going to act. They said, look, if we increase oxytocin receptors, we should see an increase in the formation and maintenance of pair bonding, and they did. They also predicted that if they introduced oxytocin receptors into the brains of meadow voles, they would see a similar change. But researchers have showed that after 24 hours or 48 hours, after being with a partner for 24 hours or 48 hours, and of course, these aren't really partners, they're just sort of like cage mates that you're stuck with because meadow voles don't form partners. They did not have a preference for that cage mate or that partner. They showed no preference, even when they were given oxytocin receptors. So that was a little surprising to researchers. We're not sure why that's the case. Other researchers are studying this nucleus accumbens and saying, hey, is it really important in the reward system? And how does it influence partner preference? There's a lot of studies out there that directly link what's happening in the nucleus accumbens with partner preferences and partner or pair bonding that happens. So that's kind of cool. The studies that are shown here, the studies that are shown here just suggest that when you stimulate the nucleus accumbens, animals have an interest in a partner. And just before, when an animal approaches his partner, the nucleus accumbens starts firing. Its neurons are going wild. So clearly, there's this connection between oxytocin receptors and the nucleus accumbens and triggering social pair bonding behavior in female prairie voles. But now there's a humongous twist in the story with respect to oxytocin that is unexpected and relatively new. So this is 2022 research. So researchers are like, all right, we know oxytocin is really important for pair bond formation in female prairie voles. And we now have this access to a special kind of editing machinery called CRISPR. Let's edit out the oxytocin receptor gene in prairie voles. And I bet we're going to see the diminishing of social bonding. I bet we're not going to see any social bonding if there's no oxytocin receptor gene. So they cut out the oxytocin receptor gene, and remember that produces a receptor protein on particular neurons in the brain, like in the nucleus accumbens. They took it out, and then they watched the behavior of these little buggers. And sure enough, they formed females formed pair bonds. They actually perform pair bonds, and they have preferences for their partner. What? That's so confusing. Oxytocin seems really important in the formation of pair bonds, but now you're telling me that you remove the gene for oxytocin receptors and they still somehow form pair bonds. There must be more to our story, right? This is where the story gets exciting, really. We don't know what's next. Researchers are going to keep looking. What about vasopressin and social behaviors, particularly thinking about vasopressin and its role in male social behaviors? They form pair bonds too. What's going on with them? Researchers have done a ton of work with vasopressin as well. And so let's look at some early studies. So early studies involved injecting vasopressin into certain regions of the brain. And they typically injected them into what's called the ventral pallidum, if I can find my cursor here, the ventral pallidum is a nucleus or a cluster of neurons that's found in the basal ganglia. That's a part of that reward system still. And by injecting vasopressin in males, prairie vole males suddenly do a whole suite of reproductive behaviors that go along with monogamy. They start parental care. They start mate guarding. they form partner preferences more easily and stronger preferences. But when we inject vasopressin into meadow voles, into the same spot in meadow voles, this doesn't stimulate any of those behaviors. Meadow voles are promiscuous. Remember, meadow vole males don't form partners, don't form pair bonds. They don't participate in any kind of paternal care. They don't guard their mates. They simply mate and leave. And adding vasopressin didn't make any difference. There are also other species differences seen in birds. So, for example, if you take field sparrows are songbirds that are territorial. They form very strong partners and will live in a territory and aggressively defend their territory from other field sparrows. Now, inject vasopressin into them, and they'll start singing more, and they'll be much more aggressive toward any rivals around. In other words, they're doing more territorial behavior. Zebra finches are colonial birds. They live in big colonies. There's not a lot of aggression going on. And if you inject vasopressin into male zebra finches don't increase their song rate. They don't increase their aggression. They just don't. And so we get different species having different reactions to vasopressin injections. So let's look at the brain, and let's look at particular regions of the brain and see if there are differences in the distribution of vasopressin receptors. And in fact, there are. Prairie voles have a really high density of one type of vasopressin receptor, and it's called the V1A receptors. So if you look at their ventral pallidums, they have lots and lots of V1A receptors for vasopressin in them. All right. When we compare prairie voles to meadow voles, let's take a look. Here's our prairie voles. Oh, they're so cute. And here's our meadow vole. And time spent huddling. Oh, okay. Yeah. When we look at their brains and we do cross sections of their brains, we actually see that prairie vole males have high levels of vasopressin V1A receptors in their ventral pallidum. And meadow vole males don't. They also don't. They don't also huddle with a partner, with a cage mate, very much. They huddle at very low rates. It turns out that this figure over on the right here looks at dopamine levels. Dopamine's a neurotransmitter that's released in this reward circuit, and it's really important for motivation and learning about rewards, etc. And dopamine receptors are very similar in both prairie voles and meadow voles. So it's not anything to do with dopamine. It has more to do with these vasopressin receptors that are just differently distributed. And interestingly, researchers said, hey, what if we added vasopressin V1A receptors to meadow voles? What would they do? So here they decided to add with this, they used a virus. This is really weird, but they used this virus and they added the AVPR1A gene. That's the vasopressin receptor 1A gene, the gene that codes for these receptors. They put it into the virus and then they put it into the brain of meadow voles, and they inserted it into the ventral pallidum. And then meadow voles started making way more of these V1A receptors. And when they expressed more of the V1A receptors, guess what they did with their behavior? They started doing social pair bonding. They started being attracted to females and pair bonding with them. So you see a change in the distribution or the expression of vasopressin receptors leads to a change in behavior. That's pretty interesting. And then researchers are like, well, these AVPR1A genes and receptors are pretty interesting. I wonder if we can see differences if we look across even different male prairie voles or if we look across different species, are there differences in the gene that codes for these 1A receptors, right? So let's look for that gene. And they found that gene. That gene is called the AVPR1A gene. That codes for the building of these receptors that are expressed in the brains of prairie voles and meadow voles, but we're particularly interested in the brains of these prairie voles, and they're expressed in that ventral pallidum area in particular. And so let's look at them and let's kind of examine them a little bit. And it turns out there are different versions of this gene, different versions or different variants of the gene. And so here's a picture of two different versions of the same gene. This is the gene AVPR1A. That's the gene that provides the code for, or the code for the building of these V1A receptors that we see in the brains, in the ventral pallidum of prairie voles, right? And the gene here is shown, the coding part of the gene is shown in dark blue. These light blue or these gray parts are what we would call the promoter region. If we look at the whole gene, though, there are differences upstream of the coding region. Differences up here, closer to what we call the 5' end of the gene, in what is known as the microsatellite region. This particular part of the DNA has repeats, has short sequences that are repeated. And there are different versions or different forms of this gene. Some individuals inherited longer microsatellite regions, and some inherited shorter versions. So there are different forms of this gene. We call that polymorphisms, many forms of the gene, different forms. There's a long version and a short version. The long version and the short version, maybe this is somehow leading to differences in the expression of this gene and differences in behavior. That's what we're kind of interested in, right? Well, let's think about this. So, by the way, this term allele just means version of the gene. So there's a long and a short version of the gene. And so researchers realized this with prairie voles, and they started doing breeding, and they bred animals so that they had animals that had only the long version of the gene and animals that had only the short version of the gene. And then they looked at their behaviors. And sure enough, there were differences. Individual differences in behavior based on the presence of the different forms of this gene. Some animals, the males with the long version of the gene, these animals were much more likely to form partners and form strong partnerships with females and do more parental care than animals with the short version of the gene. Animals with a short version of the gene are less likely to form partners, and if they form partners, they're less passionate. I don't know about that, but they spend less time with their partner. They also engage in less parental care, less building in the nest, less defense of the nest, all that good stuff. And let's look at their brains. Are their brains different? Do the brains of individuals with the long form of this gene show a different, set of receptor expression? And they do. Okay, individuals with the long form of the gene have higher V1A receptor expression pattern. So they just have more of them in the ventral pallidum. And this kind of makes sense. If you have more receptors in your ventral pallidum, you have a stronger behavioral response. You are more likely to engage in pair bonding and parental care. Pretty cool. This is a pretty cool link between a gene, the hormone, vasopressin, and the neurons that are found in the ventral pallidum. And finally, the behavior. What about other social behaviors in prairie voles? Do we see that oxytocin or vasopressin plays an important role in other kinds of like social behaviors? And so here's an article from 2016. Let's check it out. What about other social behaviors? Let's study consolation behavior. Consolation behavior, what does that even mean in a prairie vole? It's kind of evidence that animals will console each other. They recognize, animals in stress, and they will change their behavior toward them. That's what consolation behavior would be. Well, here's what these researchers decided to do. I know it's kind of confusing, but don't panic, we'll figure it out. So they have animals that are pair bonded, male and female, and they mate and they hang out together for some length of time, right? Maybe for a few days. Then they call one of them the demonstrator and one the observer. The observer and the demonstrator are separated. And while they're separated, the observer just hangs out, right? Just hangs out by itself. The demonstrator could have one of two things happen. The demonstrator could just be separated, could be in a different tank somewhere else for a little while, and then it comes back and they reunite. Or this is the stressful situation. The demonstrator is separated and then is exposed to stress. It hears a tone and then gets an electric shock, which is super stressful. So tone paired with electric shock. It soon starts to learn that the tone means an electric shock is coming. It has this experience of stress, and then it's returned to the same chamber with the observer. And now the researchers are like, hey, what happens after the reunion? What's going on? So here's a picture of what's called allo-grooming. That means other grooming someone else. These animals groom each other frequently, but is the observer more likely to groom the demonstrator when the demonstrator is stressed out? That's really what it amounts to. So let's take a look. Before and after means before and after the separation. And this is separation without the stressor, and this is separation when the demonstrator was stressed out by the electric shock. Let's look at what happens. So there's some allo-grooming that happens all the time, and this is allo-grooming by the observer. This is the little guy who knows nothing about the stress. Okay, so they do allo-grooming. When he comes back, they do maybe a little more, but not significantly more when there's no stress. But when the demonstrator comes back stressed out, look at the difference in allo-grooming behavior. That observer notices the stress in the demonstrator and responds to it with more grooming behaviors. Right? By the way, this behavior is not seen in meadow voles, period. They've also tried, they also noticed a few other things, and this is really interesting to me. They noticed that kind of matching behaviors. The observer mirrors the behavior of the demonstrator, kind of matching the anxiety behaviors of the demonstrator. This is called emotional contagion, So like if the demonstrator is stressed out, the observer is stressed out. And that's shown in these graphs down here. I can't see them very well. Let's see if I can get a feeling for them. Okay, so this is before and after, and we're looking at self-grooming, right? The observer, before and after, what happens is the observer essentially mirrors what the demonstrator is doing. So after the demonstrator experiences stress, it does more grooming. That's a stress relieving behavior. It does its own grooming. And guess what? So does the observer. The observer's copying the demonstrator. And they did some tricks to this poor demonstrator. Once the demonstrator comes back, they play the tone. And they don't follow the tone with the electric shock, but they play the tone. And what does the demonstrator do? It freaks out. It freezes. Guess what the observer is doing? The observer knows nothing about the tone, but guess what the observer does? It pays attention to what the demonstrator is doing, and it does more freezing. When the demonstrator freezes, it freezes. Is this behavior empathy-based? Are these prairie voles consoling each other? Are they um, are they showing empathy? And what's the mechanism? Is there any kind of mechanism that goes with this? Yeah, that's the question researchers are asking. Oh, by the way, if we look at the observer in response to a stress state of a demonstrator, that is a mate, a sibling, or an unrelated cage mate, you see allo grooming increases. So here's a mate or a sibling. But guess what? Allo grooming doesn't increase when there's a stranger who's stressed out. So if the demonstrator is just some stranger they've never met before and it's been stressed out and it comes to the observer doesn't recognize the stress of the stranger or doesn't respond in the same way to the stress of the stranger. Isn't that interesting? Researchers are really interested in like what's going on neurologically with respect to consoling behaviors. And they think oxytocin plays a role. So it turns out that there are a lot of oxytocin receptors throughout the brain. There are different places in the brain where oxytocin receptors are located. One of them is that nucleus accumbens. One of them is called the anterior cingulate cortex, which is actually in a different location in the brain. It's near the medial frontal lobe. It's involved in emotional regulation, bridging cognitive and emotional processes. So it kind of processes cognitively and emotions and it kind of organizes them. And it turns out that they decided to block oxytocin from different areas of the brain and then look for this consoling behavior. And they could block oxytocin in the nucleus accumbens, they could block oxytocin in the PLC, which is another area of the brain, and had no effect. But when they blocked oxytocin receptors, or they blocked oxytocin action in the anterior cingulate cortex, then the animals can't console anymore, or they don't console anymore. So the presence of oxytocin in the anterior cingulate cortex seems really important for this consoling behavior. But injecting, oxytocin antagonists anywhere else didn't seem to matter. That's kind of the research that's happening now. What about humans? Can we, is there a connection between our oxytocin levels and social behavior, or what we call social cognition? A lot of research into this, particularly because researchers believe, hey, if we can find these interesting links between social behavior and or the lack social behavior and oxytocin, or the lack of oxytocin, then maybe we can find treatments for diseases that are associated with like changes in social behavior, or unusual social behaviors. Okay. So let's think about it. Zhang et al. Just published 2024. This is an article coming out of China. Has a lot to say about humans and and oxytocin in particular related to social cognition. And they did an experiment. So you can read this if you want to, but they did an experiment that is pretty interesting, and it looked at the presence of oxytocin or vasopressin and morality. Yeah, but it was kind of awkwardly done. I mean, this is a study. So here's what they did. They gave students, of course, there were 180 students in this experiment, 90 males, 90 females. They gave them one of three things. They gave them an intranasal spray. So they did a little spray up their nose of a placebo, of oxytocin, or a vasopressin. And then they gave them some moral tasks, right? Actually, what we would call a moral emotions task and a moral judgment task. And they were kind of weird. Here's what they did. They showed them I don't know if it was a comic strip or if it was actually like a video. of figures depicting moral scenes, and in the moral scenes, um, the actors were either deliberately harming someone else or accidentally harming someone else. And then they had the students imagine themselves as the actor or as the victim of being harmed, and then rate their emotions. Like, how'd you feel? Were you ashamed? Were you annoyed? Were you guilt? Did you feel guilty? For the moral judgment task, they actually gave them scenarios, and then asked them if they would endorse those actions. And I think the scenario they had them do was a lot like that. Train switch action where you've got a train going down a track, and it's either going to kill one person or it's going to kill 5 people. You maybe know the one person, you don't know the 5 people. What would you do? You're intentionally harming one person to help other people. And they asked them, would you endorse this? Do you think this is a good idea? And people who are on placebos, vasopressin and oxytocin acted differently. Vasopressin and oxytocin were vasopressin and the placebo effects were no different. But here's what happened when you were given oxytocin. It increased your feelings of guilt and shame for intentionally harming someone. That was in the moral emotions task. And they lowered their endorsement of intentionally hurting someone to help others. In other words, they're like, I don't think I would intentionally do that. Even if I were going to help someone else, I don't think I'd intentionally hurt. I don't think that's a good idea when you had oxytocin. So oxytocin may be important to stimulate moral or pro-social behavior in humans. And researchers are really interested in this because they're interested in like the therapeutic potential for, like I said, disorders of social behavior. Researchers have looked at a whole suite of different things with respect to autism and schizophrenia. And this is what does that stand for? It's the having drugs, but with autism, for example, what does oxytocin do? What does vasopressin do? For people with schizophrenia, what does oxytocin do? What does vasopressin do? Can these be therapeutic? For people who have autism, or people who have schizophrenia, or people who are suffering from being addicted to drugs? That's a really interesting question. And in conclusion, we can say that social behavior is really complex, and genes, neurology, and hormones interact to contribute to social behaviors. All right, so we're right here in this little diagram. We're somewhere up here. An individual's genotype can influence its behavior. Notice all the other factors that can influence the cat and the dog's behavior here. We're really only right here. And so we're going to continue thinking about the causes of behavior. In this case, we looked at hormones, the hormones oxytocin and vasopressin, and how they seem to influence pair bonding behavior in a little tiny prairie vole, and how they may influence human social behaviors or moral behaviors or behaviors related to trust. It's a continuing story, and it's fun and exciting. And that's all I have for you. So I'm going to quit my live streaming here, and I think that's it. We'll see you later. Bye.